exam. The date of the final exam is not planned yet, but it should be something like 15 days after the end of the courses. And so there will be a final exam. And I will explain you at the very beginning of next week. I don't know if it's going to be possible, but I would like to include part of the evaluation, a work that you are going to do in small groups of 4 or 5 students, a very small work, which is a kind of synthesis about one particular point of the lecture. So I will give you more information on this on next Monday in 1 week. Okay, so do you have other question? Otherwise, we can start. It's okay for everybody? Okay, so I share my screen, my slides, and so if you have any question, I can swap to the whiteboard and explain you everything. So what are we going to do in this BP10, which is called molecular biology? So molecular biology, this is the name of the teaching unit, but we are going to go a little bit further about molecular biology. So this is a list. So I try to, wait, I'm going to try to, so you see the red dot, or yes, is it okay?
Yes, it's okay.
Okay, so this is the list of the concept we are going to some of them really in depth, and the other one we are going just to have a few information of that. So today we're going to look something about the structure of the genomes because it's quite important. So you will see there is a lot of information. For me, it's important that you have this in mind, because once you are going to work on specific species, the structure of the genome is quite important in order to develop molecular biology tools and things like that. So we're going to talk about that today. Tomorrow, and yes, mainly tomorrow, we're going to talk about another level of organization of the genome, which is at the level of chromatin. So we're going to see that in today and tomorrow. And then we're going to spend a lot of time about this, what is called the central dogma of molecular biology. So can you tell me if you know this sentence, the central dogma of molecular biology? Have you ever heard about that? Can you tell me? Do you have an idea about what it is?
No, I don't think I've heard of that before.
Nobody heard of, you can talk, don't worry, we are here to discuss.
I think I have heard of this.
And what do you remember from that?
I think it's kind of
Is that the process in which
the expression of a gene?
Okay. So when you speak, one person at a time, at one time. So you were saying?
Can I speak? Can I continue?
Yes. Okay. Go. Speak.
So if I remember correctly, the central dogma of molecular biology is explain of the flow of genetic information. So it's from DNA to RNA, and then from RNA to the functional protein.
Okay. So it is this. We are going to see that. So I'm going to explain it back again very quickly, but we are going to spend a lot of time on this central dogma of molecular biology. So as was saying, sorry, I don't remember your name, one of your colleagues, it's the concept of information flow from molecules to molecules. So the information is stored in DNA in terms of genetics. Then the information flow from DNA to RNA. This is transcription. And then the translation of the RNA gives rise to the proteins and then the function. So the central dogma of molecular biology, when it was discovered or when it was proposed, let's say, it is something like 40 or 50 years ago, a little bit older even. So they were saying it's Crick and Watson. We're going to come back on that later on. But that the information was stored in DNA, then was going from DNA to RNA, and then from RNA to protein. So this is what we call the central dogma of molecular biology, which is information flow. But you will see that this central dogma is completely all right. It's perfect. There is no problem, but it is not complete at all. There are plenty of other relations in terms of flow information. And this is what we are going to see in the next lecture. So we're going to start from what we know from chromatin structure and genome structure. I will present you the initial central dogma of molecular biology, and then when we are going to study the regulation of genome expression, the transcriptional regulation, post-transcriptional regulation, and we're going to see that in different cases study and exercise, you are going to see that from the initial central dogma of molecular biology, we can update. This is CDMB, means Central Dogma of Molecular Biology. So from all the case study and information that we are going to see, we're going to spend at least 9 hours on this point. We're going to make an update on this central dogma of molecular biology with all the data that we have in 2022 now. Okay, so we're going to start from a historical point of view and then increase the complexity. And we will finish the lectures next week with some aspect quite technical about how do we sequence genome nowadays, depending on the species, depending on the data that are available already. And also we are going to see some genome-wide approach in order to identify gene expression profiles in different species. Okay, so basically this is all the main points we are going to see in this BP10. And you see that there are case studies and exercise. So we will have, I have prepared for you an exercise on gene regulation, and we will see plenty of molecular biology technique through this. Is it okay for everybody? Okay. So, concerning the recommended general literature, so there are plenty of publication. The problem is that molecular biology, how can I say, it's not a science in terms of scientific publication. What I want to say, saying that, it means that molecular biology is a kind of tool if you want to study physiological processes or development of plants or animal and so on. You will find plenty of information of different aspects of molecular biology in scientific publication, but there are two books that are a very good summary, and let's say this is what has to be known in molecular biology. So those books, you know them. As far as I remember, I think that they are in the library at USTH, both of them. So you have, it's a very good synthesis on a lot of aspects of molecular biology, mainly in animals, with a few aspects in plant science. So these books are, you can have some parts of these books online or in the library. They are from 2014 and 2012, but they are really up to date in terms of processing. And you will see that a lot of images that I used in this PowerPoint slides, they are coming from these two books because they are really nicely made. Okay, so it's just after, if you want to have more information on specific aspects, you can ask me and I can give you more information on this. Okay, so this slide is just to show you what can, sorry, I have to check this. Okay. So, this is in the Oxford Dictionary of Biochemistry and Molecular Biology. This is the definition of molecular biology. So what do you think about that? They say that molecular biology, it's a science that studies the chemical and the physical basis of biological specificity and variation in gene expression, structure, and interaction. So do you see that? What would you say if you compare molecular biology and biochemistry? Do you have an idea? Actually, it's the word molecular biology comes from the name is coming from the discovery of DNA by Crick and Watson in '68. And this aspect, what we call molecular biology, basically it is the biochemistry, the global and integrated biochemistry of DNA and the partner that are interacting with DNA. So, for example, we talk about molecular biology when we talk about gene expression, but gene expression is the result of the activity of some enzymes, RNA polymerase and a lot of them, and the molecule which is red is DNA. So basically, molecular biology, you can consider that as the biochemistry of DNA, RNA, and some kind of protein. So we're going to see different aspects of the genome structure today. So, slide. First of all, we're going to talk about the size of the genomes in different living organisms. So in this slide, you have in the x-axis the genome, but in haploid genome, which means only one N, okay? So it's expressed in 10 power 5, 10 power 6 nucleotide. So it means that here 10 power 6, we are 1 million base pair, 1 trillion base pair, and you've got different type of living organisms. So you've got plant. It's almost all type of organism, living organism on Earth. So coming from the less complex, the Archaebacteria, you see that the genome size is very, very small. It's only 1 million base pair in general. And then you can have some organism like protozoan. You can see like the amoeba, it has an extremely important, very big genome. It's something like something like 10 power 11 base pair. And it's even bigger than human, frog, shrimp, or some plants, okay? So you have different size of genomes. So they are classified according to their phylogenetic relations. So for example, on the top row here, we've got all the mammals, the birds and reptiles. The little yellow bar is for human. So you see, for example, that in these mammals, birds and reptiles, the genome size range is something like 1 log, okay? So between 10 power 9 and 10 power 10 base pair. So the range is quite small. And then on the opposite, you have, for example, if you take the protozoan genome size, the range of sizes, they lies from 10 power 6, something like 5, 10 power 6, to something which is almost 10 power 12, okay? So you see that in this class of living organism, the difference of the size of the genome is extremely diverse. And there is a kind of reason from that. It's that, but it doesn't make sense for all of that, is that the diversity of protozoan is extremely wide. And this is a very, very old organism in terms of evolution. So one of the theory is that this kind of species of living animal, protozoan, they are so many, they are so old in terms of evolution. There are so many genome evolution that the range at the end of the genome size is extremely wide. Then in plant, it's a little bit the same. There is something which is very interesting in plant, is that the oldest plant in terms of evolution, they are not here. Some algae are very old. And then, so you see Arabidopsis is one of the model in molecular biology or in development, in science. This is a very small genome. Okay, it's 10 power 8 base pair. And this is why it has been chosen as a model organism for plant, because the sequencing was quite easy. But it's a dicotyledon plant. And then you have here wheat, which is a monocotyledon plant. And monocotyledon plant appears later in the evolution. So the difference in the range of the genome size is also related a little bit to the story of evolution of this organism. And there is something that is not written in this picture. If, for example, some trees, like pine trees, the genome is even bigger, and they are almost the bigger genome in the living organism. So, okay, so this is completely descriptive. You've got the ranges of the genome size according to different classes of living organism, but there is one conclusion from that, is that the genome size, this is what is written. Oh, sorry, there is somebody else which is entering the class. 1 second. Okay. So the conclusion, if you compare the genome size and the complexity of the organism, you will see from this slide that there is no clear relation between the size of the genomes and the complexity of the organisms. Okay? Because we can consider that, for example, an amoeba is extremely less complex than a wheat plant or than a human or than a frog or a shrimp as well. So there is no correlation between the genome size and the living and the complexity. But there is a kind of relation with their age. So this next table kind of synthesis on the data we have. So it's very basic data. For what we call the model organism. So in biology, we need to use model organisms, and we will come back on the definition of what is a model organism. And so you have here the most commonly used organism used in the labs to study different things. So you see, for example, so for plant doing microbiology, Escherichia coli, then the yeast, Saccharomyces cerevisiae, the roundworm C. elegans, which is a model for animal development, then the model plant Arabidopsis thaliana. I'm sorry here, there is not the rice, but rice Oryza sativa is also a model plant. Then the fruit fly, Drosophila, zebrafish, which is a model for the fish, then a model for the mammals, Mus musculus, the mouse, and Homo sapiens, ourselves. So what is interesting is here, you see, we have the size. So one important thing is this little star. I put back my pointer. This little estimation of the amount of highly repeated DNA sequences that are not present in the genome database. So I can explain you a little bit this before we go on. So when researchers work on the genome sequence of one organism, for example, one of the list here, so they can sequence the genome, and we will see at the end of next week when we will study the sequencing the genome, the technology for genome sequencing, that there is a problem when we sequence a genome. So sequencing part of the genome that contains genes is, let's say, quite easy because the sequence is not the same and we can read the DNA sequence without any problem. But what happens is when we want to have access to some part of the genome, the sequence of some part of the genome, that is composed with very repeated sequences. The problem is we cannot really have the precise sequence of these repeated sequences because the sequence, nucleotide sequence, is repeated several times, a lot of times. And at the end, during the assembly of the genome, it's quite complicated to know exactly the size of these repeated regions. But nevertheless, there are some software that can help in order to have an estimation of the repeated sequence size. And it's an important part of the genome. You will see that later on today. So the numbers that are listed in this first column here, it is an estimation, obviously, because we don't have the precise number of the repetition of the genome size that includes the repeated DNA sequences. So if you look from E. coli to Homo sapiens, which is the bigger genome in this table, the fold of increase of genome size, it's something like 1,000, something like that. So a million, let's say the range of million base pair for E. coli and the range of a billion base pair for Homo sapiens. And in between, we have different things. So genome size extremely diverse. One point is it seems that the size of the genome is increasing with the evolutionary process. But if we analyze precisely the content of these genomic sequences on all these organisms, and if we try to count the number of genes, this is the result of the number of genes. It's not precise, that are contained in every organism that are listed in this table. So you see that there are no clear relation because we have a 1,000-fold increase in the size, but we don't have a 1,000-fold increase in the number of genes. It's completely unconnected. So nevertheless, there are some tendencies that we can see. So if we cut this table here, the first two lines, they are unicellular organism. Escherichia coli is a bacterium, Saccharomyces is a yeast. So prokaryote and eukaryote. Okay, but unicellular, both of them. The genome size, okay, let's say is 3 or 4 times bigger, one compared to the other. And the number of genes, so what we call number of genes is the number of genes that are coding for a protein after translation of the mRNA. It's not almost the same, but not so much different. This is not the double. So you see that basically to have a living organism, prokaryote, something like 5,000 genes are enough. 5,000 genes, you have all the machinery that is able to do primary metabolism, the DNA replication, protein translation, lipid biosynthesis, and everything. So with a set of something a little bit less than 500 genes, you have a living organism. So the two first line, unicellular organism, something about 5,000 genes annotated, maybe more. Then the rest of the rows of this table, so they are all pluricellular organisms. Okay, C. elegans is a model for multicellular development. Arabidopsis, a plant, Drosophila, a fly, a fish, a mouse, and human. And what you see here is that, okay, the genome size are bigger, But the number of genes, if you compare this little round worm, and C. elegans is a worm that is 5 millimeter long maximum. It contains 20,000 genes, and it's almost the same as a plant, as a fish, or as human. So when we start to study the number of genes that are coding for proteins, mainly proteins, from pluricellular organisms, even if we have very different genome size, the overall number of genes is almost the same. Okay, so we can have what we can say from that okay, there is no strict correlation between the genome size and the number of genes. So what it means? It means that for Homo sapiens, in this table is the organism biggest genome, 3 billion base pair, 30,000 genes. You have Arabidopsis thaliana, which is only 200 million base pair with the same amount of gene. So all the DNA that is not present in Thaliana but present in human is DNA that does not code for proteins or things like that. But its DNA, it's important for the considered organism. Okay, so if we make a summary, there is no correlation between the size and the complexity of the organism, and no correlation between the size of the genome and the number of genes. So this is a kind of global vision of the situation. Is it okay for everybody? You can tell me.
Yes, it's okay.
It's okay. So I was telling this to you, no correlation between genome size and number of genes. We can model this a little bit. So in these two graphs, they are quite simple to understand. So we got on the left analysis of the coding DNA, and on the right analysis of the intergenic DNA. So what we call coding DNA will be the DNA that is going to be transcribed in RNA and then translated into protein. So what we can call a canonical gene, okay? And intergenic DNA, this will refer to the DNA that is in between genes unit, okay? We're not going to include intron sequence in this intergenic DNA, but we will see that later. So on the left, to summarize, the part of the genome that is coding for protein, and on the right, what is not coding for protein, and in between the genes. So the axes, they are the genome size in both Y and X axis in megabase. So what does it mean? For example, 10 power 0 means 1. It's 1, 10 power 0. So here we got a genome size of 1 megabase, okay? And 1 megabase also here, 10 megabase, 100 megabase, 1000 megabase, and so on, okay? So this will be 10 power minus 3 will be a million base pair, not a mega, a KB, sorry, and so on. So do you understand the table? So we're going to see just for the coding DNA, and I will explain you for the intergenic sequences. There is a color code that is in the little box here. So from very old, so this is virus for prokaryotes. So virus for eukaryote, prokaryote, unicellular eukaryote, animal nuclear genome, and land plant nuclear genome. Okay, so this is the different code, color code that correspond to this dot. So how to read this graph? For example, if we take this spot, the green one here, it's coming from, the data is coming from a land plant nuclear genome, and it's almost centered to 10%. Okay? So what does it mean? it means if you look at the size on X of the genome of this plant, so this plant will have a genome of, let's say, 5, 10 power 3 megabase, okay? But this plant, this is the total genome size, and it contains 10 times less megabase in terms of coding DNA. This is why this green spot is centered on this 10% diagonal line here. So do you understand what does it mean? So if we look at, there are 3 lines that are that have been drawn to show you the 1%, 10%, and 100% cases. There are obviously others. What does it mean? It means that every organism that will have a spot on the 1% line, this hashed line here. For example, if we take the spot here, it will mean that for a genome, which is 1 megabase, okay, 10 power 0, it's 1 megabase. It means that this organism has 10 power minus 2 megabase. So it means 1% of 1 megabase coding sequence. Do you all understand this, these graphics? You can tell me, because it's quite difficult to understand.
Understand.
Tell me if you understand, because I know to interpret it, but it's okay. So, okay, you can use the thumb up to say if it's okay or not okay. So, you have to be very careful when you see the number on the y-axis and the number of the x-axis. So 1% will mean, for example, for 10 power minus 2, it's 1% of 10 power 0. So if we had organism with spot all along this 1% line, it means that the genome content of this species in terms of coding DNA represent 1% of the total genome size. Okay? And if you look at this part, this line, which is 100%, what does it mean? This part, 100%, for example, you've got this spot here. So for this species in which the genome size will be 1 megabase, okay? So 1 megabase, you see, look at the type of species we have. It is the brown and kind of blue. So it's prokaryotes and eukaryotic DNA viruses. Okay, so for this kind of species that have a genome size of 1 megabase, they contain 1 megabase of coding DNA. So what does it mean? It means that this species, their DNA content, their genome content is 100% coding DNA. Okay? So if we go a little bit further in this analysis, we're going to look only this part of the 100% line. So this part, this is the green spot, eukaryotic DNA virus, and the purple, I don't know, purple black spot, which are bacteriophages. Bacteriophages, they are virus for bacteria, and DNA virus, they are virus for eukaryotic cells. What you should know is that the genome sequence of this, because a bacteriophage is basically a virus for the bacteria. So we are in front of two very specific living organisms. I don't know if we can say living organism for viruses, but we have two types of virus. one for prokaryote, the other one for eukaryote, for which their genome size, and they are very small, okay, their genome size is 100% composed by coding sequence. And this is all the strategy of the virus. The viruses, they need to have a genetic, so they have either DNA or RNA for genome information, but the size of this DNA, in the category of the DNA virus, the size of the DNA is very small. It can be only 5 kb or 10 kb, very, very small size of DNA. But all the process of the structure of the DNA in the viruses is that this small bit of RNA or DNA should have the maximum information possible. In other words, a virus is extremely clever in how to store the maximum information in the smallest DNA amount. And this is why we have this part of the graph where you see that the totality of the genome, 100%, is composed by coding DNA sequence. And then when we go a little bit further up in terms of evolution, so we are the orange one, we are still in unicellular eukaryotes. So it's going to be, for example, the case of yeast. We are not at 100%. You see that little by little, there is less and less part of the genome size that is composed by the coding sequence. And then if we go for animals and plants, it's even worse, and we go on, we go on. So it means that higher, extremely evaluated species, they have more and more, less and less, sorry, part of their genome that is coding for DNA that is going to code for protein. And the graph on the right is exactly the opposite, because on the left, we're talking about the ratio of coding DNA related to the genome size. The graph on the right is the ratio of non-coding DNA, intergenic DNA, related to the genome size. So, for example, if we see, we can take the case of these three species on the left, which are red. So red, it means that they are animal nuclear genome, these three species. They are on the line of 1%, which means 1% of their genome size is composed by coding DNA. But if you look at the same three species there, you will see that the intergenic sequence is going to be almost the same as the genome, because we're going to 1%, and here it will be 99%. You see, we are not at 100% completely. Okay, so both of them, we can say that they are mirror or opposite of one of each other. Okay, but these graphs are quite interesting because we really see, in terms of evolution, we see it better here. So very less evaluated virus, almost only coding region, and then we are starting to have unicellular prokaryote, and then we are having there most of the species we are working on, they contain something like 80 or 90% of non-coding DNA. Okay? If you have any question, do not hesitate. Huh? So there are strange things anyway. I show you this on this slide because there is no correlation, no logic comparison or biologic comparison, but there are strange things. If we look at flying vertebrates, okay, flying vertebrates, you have two pictures of the most common and the only flying vertebrates, the birds and the bats. Okay, birds, they are extremely closely related to how can I say, Okay, they are not mammals at all. I forgot the word. Anyway, so this is flying species. They are vertebrate, and the bats. A bat is a mammal. Okay, it's very closely related to human anyway. The bats. Both of them, they have a converging evolution for flying. Okay, so that's a converging evolution for mobility, for flying. But if we look at the genomes, the composition of the genomes between these two big groups of living organisms, we can realize that on birds, 10% of the genome is composed by repeats. We are going to talk about this DNA repeat later on this morning. So it means that the genome of a bird, you've got plenty of genes that are encoded for protein. There are pieces of the genome that does not code for anything, and there are a small part of the genome that is composed by repeated sequences. In birds, the repeat sequences cover something like 10% as an average. If we compare to other mammals, to the different mammals, they are spanning from 50 to 30 to 50% of repeats in mammals. So 10% in birds, and we found back 10% also of repeat DNA sequence in the bats. That's really strange. So there is a very nice paper. It's a little bit old, but it's very interesting. It was published in the very, very prestigious paper Science in 2014. And it's a kind of strange observation. It means that normally mammals are between 30 and 50% of repeated sequence in their genomes, but we found the same amount in birds, and the only correlation that we can have between those two kinds of species is their ability to fly. So the authors, Zhang et al. In Science, they suggest that there is a convergent evolution that the flight creates. The word creates is quite complicated. So it means that there was a selective pressure on the genome to shrink in the case of animals that were supposed to fly. The authors of this paper, they do not go on a little bit further on this explanation because it's quite complicated to understand what kind of selective pressure you can have in order to have the genomes to shrink because the animals are flying. But anyway, this is a constatation that is very interesting, is that the size of some part of the genome, in that case, it's the repeated sequences, they share common behavior to the behavior of flying, for example, in that example. It's okay? Tell me if you want. Okay, let's go on. So, we are going to go a little bit more in depth in 1 second, because I don't see the I need to move this. 1 second. Okay. Okay. So in this, I'm sorry, I forgot to put the reference, but I will find you back. So this slide, there is this picture, and it's very interesting. I hope you can see it very well. So we have a locus, a piece of genome, which is 50 kb here. So you see all the endpoints, they are 50 kb, 0 kb, so it's a way to see the size. So in this study, they take, it's very important to understand this drawing is a schematic reconstitution of the conclusion. So it's a general aspect. So what they did in this study, they took 50 kb genome fragments from different species in different parts of the genome, and they analyzed the content in terms of genes of this 50 kb fragment. So they did that on the first lane, A is on human. The second, B, it's Saccharomyces cerevisiae, so yeast. The third one, it's Drosophila. And the fourth one is maize, the plant. And the last one, it's the bacteria E. coli. Okay, so we've got a mammal, a yeast, an insect, a monocot plant, and a bacteria. Okay, so major representative of all the genera in life. And what they did is, so it's computer-based analysis. They checked in plenty of 50 kb fragment, or they choose 50 because it was just, they could have choose 100 kb or whatever. And this is the average of the output of the result. So the color code is this. Oh, sorry about that, but the difference of the color is very nice. Anyway, in dark red, so for example here, it's what they call a gene. In orange here, for example here, so dark red and orange. Orange is intron. In orange on the top, only in human, they wrote that, it's written pseudogene. I will come back on that later. And in blue, it's what they call genome-wide repeats. So all these little blue boxes, they are repetition of sequences of DNA that you can find repeated everywhere in the genome. And where there is a T, it's not, for example, here, it's the gene that are encoding for tRNA, the transfer RNA. So what you can see from here is it's another way to see, it's a more visual way to see what we talked about a little bit earlier. It's the gene density according to the species what we are studying. So if we take at the prokaryote E. coli here, you can see that over the 50 kb, plenty of fragment of 50 kb that has been analyzed. Most of them, most of the time, it's on 80% is composed by genes that are coding for protein. So it means that you don't have, so first of all, you never have introns in prokaryote. Okay, so this is why we have only red boxes. And you see that the density of gene is extremely high. All the genes are close together, one by one. We have, okay, because we have also some genome-wide repeats, but they represent a very small portion of the genome. At the opposite, if we come back, so I come back in this drawing, for the plant, so we have wheat here, maize is somewhere there, it's an extremely big genome. And if we look at in detail what is the structure of this 50 kb part of the genome of maize, you see that among 50 kb, if you look at the scale, there is, let's say, 2 or 3 kb, so less than 10%, it will be something like 5%, of the sequence that codes for protein. Here we have ADH1, which is alcohol dehydrogenase. But anyway, that's an example. But you see that, for example, in this 50 kb region of the maize genome, we have only one gene, and all the rest on the right and on the left, it's completely composed by DNA repeats. And this is the part of the genome that contain these repeats that explain the very big size of the genome of this species. And then we have, okay, different situation. One which is interesting is the Saccharomyces cerevisiae genome structure. So it's eukaryote, unicellular, small genome, the same amount of gene almost as human or plants. You see that the density of coding sequence is very important. It's a eukaryote. It contains introns, but you see that the density of introns They are extremely small. In other words, the size of the introns in Saccharomyces cerevisiae genes, they are smaller than in human or the eukaryote, smaller and fewer introns. So basically, you have mainly coding sequence, and we have Tai2. Tai2 is we come back later on that. It's a retrotransposon sequence, but which is repeated. Okay. So the summary of this. Okay, prokaryotic, extremely high density of coding sequence. Very basic eukaryote, the same, okay, but you have more repeated sequence. Extremely big genome size eukaryote like maize, a lot of repetition of DNA and a few genes. And human, it's a little bit in between. There is something that we have very often in human. It is what we call pseudogenes. Pseudogenes, it's don't be afraid about that. This is a gene that during the evolution stopped to be functional. In other words, it was a gene that has no use at all, and little by little it has been mutated. So you have kind of, you may think that it's a gene, but there is no transcription or no translation. So, and you see in human also, you have exon, you have coding genes, you have introns and exons. You have pseudogenes, and you have a lot of repetition also, and the genome size of human, it's very important. It's 3 10 power 9 base pair. Okay, so why I wanted to show you this is I show you on the table that you have an increase of the genome according to the evolutionary process, but we can have detail on what is increased. So technically, what does it mean? It means that the increase of the genome size is coming not from an increase of the number of genes or the size of the genes, but it's coming from an increase of the repeated sequences in the genomes. Okay? Excuse me, there is somebody that wants to enter the room. Is it okay? Okay. Okay. If you have any question, you can ask me.
Can you say again the conclusion with
the conclusion on that, on this slide?
Yes.
Okay. So, but it's a general conclusion. So we see on the other tables that during the evolution process. So I come back in another diapositive here. So, for example, if we take this line for the plant, Arabidopsis is a dicot. Wheat and lily, they are monocot. Okay. In the evolution, the dicot appear really earlier before the monocot. In other words, monocots are more evolved than dicots. Okay, and if you look at, because this is a good example, the plants. If you look at the genome size of a dicot plant compared to the genome size of monocot plant, you see that monocot genomes are really much more bigger than dicot plants. After, in terms of evolution, human is considered to be more evoluted than, for example, fish or Drosophila, and you see that there is an increase in the genome size. What we see in this detailed view, we see that the increase, if we follow the evolution process, so for example, the less evaluated, I don't like this word, the evaluated is E. coli, and let's say the most evaluated in this table is maize in terms of evolution. So from this way of looking at the genes and the structure, you can see that the increase of the size of the genome between coli and maize is mainly due to the increase of repeated sequence, but not genes by themselves. And this, we found it back in this table. So if you see, you remember this table, we have, for example, for the plant Arabidopsis thaliana, we've got 30,000 genes. It's almost the same, one per one to Homo sapiens, but the size of the genome is totally different. Well, it's very complicated to compare plants and human, but you can compare, for example, C. elegans, which is an animal, multicellular organism, and Homo sapiens or the mouse. You see that the difference on the size of the genome is huge, but the number of genes is not very important. And this increase in the genome size is mainly due to the increase of repeated sequences. Is it more clear now? It's okay? So one conclusion would be that this is what is written on the right, is the more simple is the organism, the less gene repetition you have. So it's a way to explain the dynamic of the size of the genome when we compare to evolution position in the... It's okay, do you have a question or we go on? Okay. So we're going to see a little bit of specificity of the human genome. Just a quick reminder to explain you why and how and when the human genome sequencing was done. So it was a very challenging research program that only consortium of laboratory could achieve, because it was very expensive and extremely fastidious. So the aim of the sequencing of the human genome was very clear at the beginning. Technically, it was very difficult. The aim was to sequence the 3.2 10⁹ base pair of the human genome. The objective was to identify all the genes that are coding protein. And this was made possible because I don't want to come back in the time, so everything has been started in the 90s, sorry. And why it has been possible at that time? It's because of the possibility of using very archaic bioinformatic tools at this period. But you have to keep in mind that the sequencing of the human genome wouldn't have been possible without the use and the development of specific bioinformatic tools for genome analysis. And from that, so it was a very important step in genome sequencing, because nowadays you can sequence the genome of a new human, a new individual, in less than a week. It doesn't cost anything, but the first genome, the first draft of the genome, it took years, something like 8 years to have a raw sequence. And it was mainly because all the bioinformaticals were not present, and they had to be developed specifically for this sequencing project. So there's something, but we won't talk about that in this lecture because it's quite complicated. There was, for the human genome sequencing, there was 2 parallel programs. Both of these programs, excuse me, I need to stop the, wait, 1 second, because, so, those 2 programs, sorry, I didn't, so there was the Human Genomic Genome Project. The kickoff of the project was in the 80s, and it's very important to understand that it was a public consortium to coordinate plenty of different labs from different countries, and they share different chromosome for sequencing. For example, France did the chromosome 40, and very important from this Human Genome Project, the data were completely public to the scientific committee. So this was very important for all the researchers in human genetics. And in parallel, it's well, okay, the company was founded 8 years after the Human Genome Project. The company is called Celera Genomics, and you may know this the CEO, which is called Craig Venters. And we won't talk about that in the frame of this lecture because it's more complicated in terms of ethics. So this private company, they sequence a lot of human genome, but the data are not available. And some researchers, they are wondering what they want to do clearly with this. But anyway, so what is important to understand that this human genome sequencing was something that was extremely difficult, and it has been achieved by the public community and private company, and they didn't share the data at the beginning. So the result of these two sequencing program, the public one and the private one, both of them, they published the sequence of the human genome. It's 20 years ago, okay, in 2001. So you see, they were in competition, the consortium and Celera Genomics, they were in competition to first produce the draft genomic sequence of human genome. You see that they managed to publish that the same year. It's almost the same, but they published both in the same journal, and they are technically they succeed to sequence this genome at the same time. And I don't know if you know this this paper Science. Usually the articles are very, very short. It's 1 or 2 pages. But if you look for these two publications, the number of pages, it's very important. So this is 60 pages and something almost the same here. So these two publications were, so it was the complete genome sequence and the analysis of the genome sequence in human. Now there is more than 2,000 different genomic sequence from human that are publicly available. What we learn from this sequencing. So now we are talking about human genome. Human genome. So this table, it's coming from this this book that I showed you at the beginning. It's I like the way they say it's vital statistic. So it's the main characteristic of the human genome. Okay, so you've got different of parameters here. You've got explanation at the bottom of the. So we've got the size. Okay, we know the size, but we're going to see a few numbers there. So 3.2 10 power 9 nucleotide pair. The number of genes 20,000. We talked about that. What is interesting? This is the largest. So the biggest gene, can you imagine that? The largest gene is 2 10 power 6 base pair. How can you, what does it mean according to you? Do you have an idea? You don't have an idea? Okay, it means that you have, okay, I'm not going to draw. It means that one gene, so one transcription unit, okay? The biggest one, okay, maybe I will try it, I will make, wait, I'm shifting to the, okay, can you, Okay, it means that a gene is composed of a promoter, then you've got exons, intron, exon, introns, and so on. Okay. You can see the drawing on your screen? Okay, perfect. So this is what we call, okay, we call that a gene. A gene, it contains, so here you've got the promoter. So we're going to study the promoter very in depth later. And then this is the coding region. You've got exons, intron, exon, intron, and so on, okay? It means that in human, this is what we can call a transcription unit, because you have the promoter here, which is the switch, and the coding region, which is all this, okay? So what it means is that this part, so a complete transcription unit, can be up to 10 power 6 base pair. It means 2 million base pair. So what does it mean? It doesn't mean that the protein is going to be extremely big, because the protein, we're going to see that in detail later on, another day, is going to be translated only from the exons. So it means that, for example, you can have, let's say, this part, this is an intron here. This intron can be something like 15 kilobase. It's not a problem. You have splicing, so splicing is the removal of the introns. It means that a gene can be as big as 1 million base pair, but the protein won't be a big one. It's because of the size of the introns. And what is very complicated to understand is that in some cases, introns of very, very big genes, you don't have another gene, but you can have transposon or things like that, other transposon. Or you can have other DNA inside the introns. It won't affect the protein, because the protein will be the fusion of all the exons. So at the end, the messenger will contain only the exon. We're going to see that after. And the protein will be normal. But the size of the genome can be extremely, extremely, extremely big. Is it okay? Okay, perfect. Thank you. So, for example, the largest gene, more than 2 million nucleotides. So this is more the mean size of the gene that codes for protein. This is better. It's 2.7 kb. This makes sense. Okay. The smallest number of exon per gene, 1. What does it mean, this? It means that you've got in the genome, in human genome, you've got genes that does not contain intron, okay? The largest number of exon per gene, 178 exon. It means that this gene contains 178 exon. So it means 177 introns. So it can be a gene that is extremely big, but it doesn't mean also because you have a lot of exons, sorry, the exon can be very short. It's not a problem. As a general rule, the number of exon per gene, it's a mean, it's 10. In human, you have as an average 10 exon per gene. The largest exon size, 17 kb. It means that you have genes, at least one gene in human genome, where there is one exon that is 17 kb, which is huge, okay? The mean size of the exon, okay, this is the size, and you see the number of pseudogenes. That's interesting because in human, we have number of gene coding from protein, it's around 20,000 gene, and the number of pseudogene is almost the same. So you have the asterisk. They say it's a rough estimate. So all these pseudogenes are like fossils of gene duplication. We're going to talk about that later, but they are not functional at all. Then you have the percentage of DNA sequence in exon, protein coding sequence. Okay, you have this and this. And last point, and we're going to talk about that now, the percentage in the human genome of the DNA which is coding high copy number repetitive elements. You see, it's almost 50%. So from this 3.2 10 power 9 base pair of the human genome, you've got half of the genome that is composed by high copy number repetitive elements. Okay. So you have a kind of summary at different level, different scales of what happened. They take in this slide. That's the example of the chromosome 22. So they say in its mitotic conformation. So when we write the chromosome like that, it's during the metaphase. So chromosome 22 on human, it's a little bit particular chromosome because we're going to talk about heterochromatin. But basically, they make the scales like that. So this is the full length chromosome. If you zoom to this part, this is the B lane. So 10% of the, they say to you, okay, on this chromosome, 10% of the chromosome arm, this means 10%, is approximately 40 genes. They say to you, they explain to you everything. In this region, they will say to you on this bit of DNA, you've got 1% of the chromosome arm that contains 4 genes, and on this one of these 4 genes, you have the structure of 1 gene. So this is from the higher order of complexity to the lower order of complexity, the structure of the chromosome. So we can take it back from the bottom to the top. So this is the structure of 1 gene, promoter, regulatory sequence. In red, the exons. In gray, the introns. That's 1 transcription unit. So 1% of the chromosome arm contains 4 genes. And they say to you 10% of the chromosome, so it contains 40 genes. And at the end, they say to you, they explain to you that everything is, this chromosome is 48 multiplied by 10 power 6 nucleated. So it means that if you zoom inside the chromosome, you will see that very few part of the chromosome is containing coding sequence, and the coding sequence, they are only the one which are in red here that give rise to the RNA and protein after translation. It's another way to see the reduction. So this is there are 2 slides exactly the same. This one is the numbers. They are in megabase, and the next one it is precise in percentage. So you've got that's the overview of the human genome. So basically, what you have, the size of the genome. Okay, the human genome is 3.2 10 power 9 base pair, and they show you with all these boxes what are the part of this full-length genome sequence that are 4 genes. So, for example, the total genome size, you can divide it in 2 parts. Among this 3,200 megabase, you've got 1,200 megabase that are related to gene or gene-related sequence. The other part, so you see it's already bigger, the intergenic sequence. So it means that the intergenic sequence represents more than the gene or gene-related sequence. If we take, if we only look at the gene and gene-related sequence, among these 1,200, you have a very small part that is composed by gene. So you see it's less than 1/5. The rest is related sequence. From the related sequence, what do we have? These famous pseudogenes, which are genes that are not functional at all. Gene fragments, what does it mean? It's kind of pseudogene. Gene fragment would be during evolution, you have plenty of recombination, and it can happen that a gene is broken in 2 pieces, and then the result of the break is not functional, and you have like traces of gene fragment in the genome. And what they include also in related sequence to gene, they include the introns and the UTRs. UTRs, this is the 5' and 3' UTRs that we find in messenger RNA. It means untranslated regions of the message. So only for what is gene or gene related, it's less than half. What really codes protein is very, very few, and the rest doesn't, is not directly implicated in protein biosynthesis. The second part, what we call intergenic sequence, this is where it's more complicated to classify, because this is what intergenic sequence, it's going to be a lot of repeated sequence. So you have interspread repeats, other intergenic region. Then from this repeat, they divided in different class. You probably heard about lines, it means long, inverted, and so on. Sign is small, inverted repetition. So you have huge amount of this. LTR, they are retrotransposon, long terminal repeat elements. So this is retrotransposon. DNA transposon, so this is mobile DNA, and other intergenic region, okay, microsatellites, we will see that a little bit, and different sequence. So if the analysis of the genome, human genome, is very interesting, because you see that from 3,200 megabase, if we want to be very restrictive, there is only 48 megabase that is going to be directly translated into protein. So it's a very, very small part of the global genome. But all the other parts have can have a use in the life of the organism. So same slide. Instead of having writing the size of these different classes, this is the percentage. Okay, this is, I just calculated the percentage. So you see, we are repeats 40%, 44%, and they don't include the 2.8% of microsatellite. So almost 50% of repetition, DNA repetition in the human genome. Retrotransposon, transposon, quite important part. Together, it's almost 10% of the genome. And among these 50%, you have also 25, 35% of this line and sign repeated sequence. And the gene, properly said, the gene represents only 1.5% of the total genome of the human genome. Okay, do you have any question? It's okay. So just to be clear, this part of the lecture today, there is a lot of information. You don't need to know that perfectly. I mean, it's I have to explain you the structure of the genomes and the evolution of the genome, just to have you have to keep that in mind, that there is only a small part of the genome that it's composed by genes, and it's to give you an overview. There are not a lot of research on this aspect, but we talk about more on that later. Okay, so it's maybe a better way to remember it. So that's the global synthesis of all what we saw just from now. So the way to read this this diagram is like that. So you've got this axis, which is the percentage, and then there is a color code there that correspond to different categories of genomic region. So for example, in brown, you've got the line sequence, in green, the sign, the gray is retroviral-like element, DNA only transposon fossil, for example, in brownish. In green, non-repetitive DNA that is neither in intron nor in codon, okay, a big part, introns and protein coding genes in red there. So this is a way to represent the ratio of different categories of DNA sequencing to compose the 100% of the genome. So this is a nice way to show the thing. You see that, for example, line, sign, retrovirus-like, DNA transposon like this. So almost 50%. This is repeated sequences. Then you have non-repeated sequences that are occupying 30, a little bit less than 30%. And then the real coding is what we saw, it's 1.5. It's very, very small amount of genetic data. Okay. I propose you to make a break. Is it okay? There is at least, we'll put this number 50%. 50% of sequences that are contained in the human genome that are sign, line, retroviral-like elements or DNA transposon. So it means mobile element. So I'm going to talk to you a little bit about what we are not going to go in details, but it's in the frame of the genome structure. What do we call this retrotransposon that are integrated permanently into the genome and the transposon? So this little slide is just to give you a little bit of information. So first of all, you've got the signification of a line and sign element. So it's nothing very complicated. Line are long, sign are short, but they are interspread nucleotide repeat elements. So it means it's elements. The sign is a little bit different. We're going to see that just in a moment, of sequences that are repeated and that are coming from ancestor of retrotransposon. So what we find in the genome that looks like retrotransposon, there are these two classes, the line and the sign. So the big difference between line sequences and sign sequence is line are autonomous. So I will explain you what it means. It means that they can jump by themselves from one place to another one or duplicate. But the signs, they are non-autonomous. They are not able to change of position by themselves. So you see here the structure, schematic structure of this line and sign. You see they look like mRNA. There is a poly A tail there. They contain sequences that can be translated. For example, the second off is coding for the DNA, the reverse transcriptase, the polymerase. You've got an idea of the size of the element. You've got an idea of the number of copies of this element inside the human genome. And in terms of percentage, what they represent in the whole genome. So we said that all of them together, they represent something like 50%. Here, the total is 45, but we are roughly in the same magnitude of order. So retrotransposon type, we've got the line, autonomous, the sign, smaller, 100 to 300 base pair compared to 6 to 8 kb, non-autonomous. They've got the poly A tail that really shows that they were retrotransposon a long time ago. Then we've got retrovirus-like element. So next slide, I will explain you what is a retrovirus and how it can be integrated in the genome. They are autonomous, most of them. So they are characterized by the presence of two sequences that are repeated in both ends of the retrovirus-like element. Then we've got also a polymerase, which is normal because this element is autonomous. The size, okay, something like 10 kb when they are autonomous, and really shorter, 3 kb when they are non-autonomous. So look at this place, for example, the retrovirus-like element. You have two kinds of this category, the autonomous one and the non-autonomous one. The only difference in terms of genomic organization between the autonomous and the non-autonomous is the presence of the polymerase. So there are some open reading frames still present in the non-autonomous version, but it's not functional anymore. Number of copies quite important also, and almost 10% of the genome. And the transposons, what they are transposons that we called fossils. It means that there were transposons a long time ago. A transposon, I have a slide just after, is a piece of DNA that is mobile, that can jump from one place to another place in the genome. But to do so, they need to be active, and they need to have an enzyme which is called the transposase. And some of them, when they appeared a very long time ago during evolution, they are considered as fossils because now they almost are not able to jump from one place to another one. And it's written there, the autonomous one, a size of 3 kb and contain the transposase, and the non-autonomous, the transposase has been deleted, but there are still repeats. Okay, so this is just to give you information. You don't need to know that, you don't need to know the numbers, but it's for you to understand what is a Lyme type sequence. So you have to remember that it's a very old retrotransposon type, sign the same, and then you've got the transposon type of repeated sequence, and this is almost 50% of the total genome in Homo sapiens. So to talk about this transposon and retrotransposon, so we won't talk about that later on in this teaching unit BP10, because we've got a lot of things to say. You probably know, but it's more, I added this slide just to remind you what is the difference between, on the left, we've got a transposon, and on the right, we've got the retrotransposon. That's the A is transposon, B is retrotransposon. So just to remind you the way they work, so transposon are DNA sequence that can be excised. So it means cut from the genome with specific enzyme that they are contained inside this bit of DNA, inside the transposon. So there is an intermediate, which is a double-stranded DNA. And this double-stranded DNA, which is after excision, it's free in the nucleus, can jump and be inserted in another place of the genome. So basically, the mechanism is a cut and paste, no effect on genome size, in theory. Okay, because one transposon at one place will be removed from its original place and then inserted back again in another place. So there is no real increase or changes in the genome size. But it can happen because sometimes there are some kind of DNA sequence that are still left after the jumping of the transposon. But basically, what you have to remember that a transposon is a cut and paste mechanism. You cut it from one place. The intermediate is double-stranded DNA, and it's going to be integrated in another place of the genomic DNA. Whereas the retrotransposon, they work a little bit the same, except that they pass through an intermediate, which is RNA. So a retrotransposon, which is already inserted, and we will talk about the origin in the next slide of this retrotransposon insertion in the genome. A retrotransposon is a double-stranded DNA that is inside the genome of any individual. It's double-stranded. It encodes different proteins in which you have an RNA polymerase specific or a reverse transcriptase as well. So the copy of this double-stranded DNA will be done through an RNA intermediate. This RNA intermediate is going to be copied back again, copy back again to double-stranded DNA through the activity of a reverse transcriptase, which is an enzyme that is encoded and present in the sequence of the retrotransposon itself. There is no reverse transcriptase activity naturally inside the inside the eukaryotic genomes, except in retrotransposons. So double-stranded DNA, transcription through an intermediate molecule, which is RNA, then reverse transcribed back to double-stranded DNA, and then the same fate as for the transposon, the double-stranded DNA is going to be inserted somewhere else in the genome. The difference is not cut and paste, but it's copy and paste, because retrotransposon, the original copy, is copied through RNA, and then the new copy that is going to be inserted is an additional copy. So it has a drastic effect on copy number. Okay? What we have to keep in mind, is it okay for the differences? This, you should know that from what you studied before, the difference between transposon and retrotransposons. So they can have either almost no influence in genome size or a direct influence in terms of copy number increase directly on the genome size. But what we have to keep in mind, this is not the aim of this lecture, is that those mobile elements, we call that mobile element because they are able to jump from one place to another place, they are at the origin of mutation at their site of insertion. So they are really important in terms of dynamic of the genome and in terms of evolution of the gene functions. This is quite easy to understand, is that, for example, it can be either a transposon or a retrotransposon. Once it is inserted in another place of the genome, you can have, for example, if this bit of DNA is going to be inserted in an already active gene, it will be a knockout of this gene, so you will lose a function. There are other possibilities which are more complicated, is, for example, retrotransposon, they contain a promoter sequence because they need to be transcribed in order to produce the RNA intermediate. So it means that this mobile element, the retrotransposon, contains a promoter that is able to respond to inside the cell. So it means that once the retrotransposon has been copied and has been inserted somewhere else in the genome, it can knock out a gene if it falls down inside an already existing gene, but also it can activate genes that are on the right or on the left because of this promoter that can transcribe the flanking sequence on the right and on the left of the transposon. So the key point to keep in mind is this mobile element can affect the size of the genome in the case of retrotransposon, but they are also the source of a lot of functional diversity because they can produce local and punctual mutation at their site of insertions. Okay. So that's the two categories, and I'm going to tell you a little bit where this kind of retrotransposon are coming from. We have plenty of retrotransposon in our genomes, and a lot of studies showed that they are originated, all the retrotransposon that we found back in most of the genomes, they are coming from very old viruses infections and retroviruses. So here you have the living cycle, the replication cycle of the HIV virus. which is a retrovirus. And you will see the parallel that is very obvious with retrotransposon. And this is the way some retrotransposon has been generated inside the genome and never come back to viruses. So I explain you that a little bit. So retroviruses, you know them. There are several classes of retrovirus, but the genetic content is stored in an RNA molecule. So this is what is shown in blue here. So the cycle is very simple. The virus enters into the cell, it delivers its viral RNA. Inside the viral RNA, you have a reverse transcriptase. This is exactly what I was telling you for the retrotransposon. It means that the RNA, the viral RNA, encodes for several proteins, the capsid protein, different protein important for infection, and the reverse transcriptase. So this copy of the RNA which is delivered inside the cell is going to be reverse transcribed to DNA and then turned to be a double-stranded DNA. And this double-stranded DNA is going to be integrated in the genome. And from the integration in the genome, since this structured double-stranded RNA contains a promoter, this bit of DNA that contains the full genomic sequence of the copy of the RNA of the virus will be able to be transcribed. A lot of copy of the viral RNA. All this viral RNA encodes for all the protein that compose the virus, the capsid protein, the envelope protein, reverse transcriptase, and others. And everything together is going to be assembled and to create a new virus particle. So this is the classical cycle of replication of an RNA virus, okay, a retrovirus. What happened is that the retrotransposons that we found present in the genome of most of the living organisms, for now, at least a eukaryotic one, you have to consider that they are coming from, in terms of evolution, to very old viruses, retroviruses attacks. But once it has been integrated, some of the function disappeared. For example, you can have the capsid protein that is mutated by one event of mutation. And with the evolution, it means that at the end, the global information that is stored in this integrated DNA is not sufficient enough to produce all the different partners. So it means the RNA, the capsid, the envelope protein, the RNA reverse transcriptase. Some of those proteins have got mutation, and therefore the new virus cannot be assembled. So, in terms of history, retrotransposon comes from a retrovirus infection, followed by mutation of specific gene inside the retrovirus sequence that has, as a conclusion, the inability to produce new viral particle. Therefore, this piece of DNA stays in the genome, but still have the capacity to transcribe its RNA and then jump from one place to another place. So basically, a retrotransposon, if we want to really make a shortcut, it is a retrovirus that has been integrated, done loss of function of a few genes inside the retrovirus, and from that, the new viral particle cannot be produced, but the double-stranded DNA pieces can jump from one place to another. So it stays inside the genomes. Is it okay for all of you? The origins of these retrotransposons? Okay. So this is not very important. It's just to show you the structure of the genomic structure of proviruses. Retrotransposons, so you will see there is plenty of similarities, and the limes, which are very reduced version because they lost a lot of sequences from the provirus. So for example, you see the provirus characteristic sequence. In brown is the genomic DNA of the host. You've got these two LTR. LTR means long tandem repeats. there are two, one on the left, one on the right, and these are the promoters for the transcription of the viral genes. So typically, this is a provirus. It means the result of the insertion of a double-stranded DNA coming from a retrovirus inside the genome of the host. You've got this. From that, you can have transcription, production of the viral protein, and then new viral particle. Okay, so this will be the normal cycle of the virus. This is one step of the cycle. Retrotransposon, what it is? It's exactly a retrovirus, but if you see the LTR have been shortened a lot, the protein coding region is really smaller and does not contain all the viral genes. So it means it's part of a virus, but it's not a fully active virus. You see that on one hand, they still contain the long terminal repeat that is able to transcribe for the RNA, the reverse transcriptase. And the Lyme sequence, they are even more simple. It's like if you take a retrotransposon and you remove more and more sequence. So what do we found from in the Lyme? We only have the reverse transcriptase. This is the only protein that can be translated from the Lyme RNA. We've got a poly A tail in order to be translated, and this is a constitutive promoter. So we start in terms of evolution. Provirus turns to be retrotransposon with the disappearing of a few sequences. And if we reduce more and more, we've got Lyme sequence. And then those sequences, they are not really able to jump from one place to another one, but they have been multiplicated, and this is what caused the big percentage of repetition of this kind of element. So I hope that you see the relation between viral attacks, ancestral viral attacks. Million years ago, so many virus attacks, we still have the places of insertion of this virus a long time ago in terms of evolution that turned to be a retrotransposon after all. Okay, any question?
I have something I want to reconfirm.
Yeah, tell me.
Yes, so is the gene in the line always the reverse transcriptase, or is it just like in this example?
Alors, it's always the reverse transcriptase in the Lyme. The Lyme L stands for long, okay? In the Lyme sequences, you always have the reverse transcriptase, sorry. In the sign, which S sign stands for short, in the sign, the reverse transcriptase is not present. There are bits of the reverse transcriptase, but it's not active anymore. So there is, in the Lyme, if it is your question, there is only the reverse transcriptase, and it's active, functional at least. Was it your question?
Yes, that is my question, and yeah.
Okay, and in the retrotransposon, the dark green part that I don't know if you see the mouse in the screen.
Yes, I can see it.
Otherwise, I'm at a pointer red. The dark part of the green, they should have put an S, proteins encoding region. There are different proteins. Among them, there is the reverse transcriptase. Okay. And the long terminal repeat, the only one that stays in the retrotransposon, compared to the provirus, there are two. Those LTR sequences are promoter-like sequence. So retrotransposon, if you check a little bit, retrotransposon. Let's start with provirus. Provirus, they contain this LTR, so promoter-like sequence. All the viral genes, so once it's transcribed, you can have the protein translated and you can recreate new viral particles completely functional. Retrotransposon, you lose plenty of genes, you keep the reverse transcriptase, you remove one LTR. So one LTR is still present, so it can act as a promoter to transcribe this region. And the part that can encode a protein does not contain all the protein necessary to recreate a viral particle. So it cannot live outside the cell. But it's going to be transcribed, reverse transcribed, and reintegrated in another place in the genome. Okay?
Yes, I understand. Thank you.
Okay. And from this, so in terms of evolution, provirus, they can, okay, virus, they enter the cell, multiply inside the cell, and go out the cell. Okay? Retrotransposon, they are now inferred inside the genome, but they cannot move outside the genome. They can jump inside, but they cannot go to another cell or another. And the Lyme, this is the same. They are completely stuck to the place where they are. Okay. So we're going to skip this one. It's just more detailed. It's the way of the transposition of a non-retroviral transposon. So it's a transposon like a Lyme. So I just show you there. You see in the Lyme, there is in pink here, it's the poly A tail. So this is what we find here. L1 is a Lyme. So we've got in orange genomic sequence, in red the Lyme, you have the poly A tail, and you see it's double-stranded DNA integrated inside the genome. But it can be transcribed. So here we have a fully functional mRNA. There is a reverse transcriptase endonuclease that is encoded by this RNA that is going to bind its poly A tail. And this is this complex because it contains endonuclease activity and reverse transcriptase activity that is going to cut somewhere else in the genome one of the double-stranded DNA and then going to be integrated. So the site here from the beginning is a different one from the one you copy at the new position in the genome. Okay, so this is the mechanism if you want to have a little bit more detail. But it's not necessary to know that just if you're interested in this. Okay, so to finish, I show you just some data that are quite interesting. ALU sequences, it is one category of the SINE family. So it is small repetition, something like 300 base pair, but we can find them something like 10% of the human genome composition is composed by these ALU sequences, okay? And they come from the retrotransposition of a non-coding RNA, okay? And you have, okay, it's not very informative, but it's quite interesting to see how it works in this schema. So here in Y, you have time in million of years, okay? So 0 is nowadays, and this is 60 or 80 million years ago, where this non-coding RNA appeared, okay? And because it was able to jump and to duplicate, because it's a retrotransposon-like, so it's copy and paste and not cut and paste, those sequences in human, starting from 1, nowadays we've got 1 million of copy. So I don't know if you see in the drawing, this is all the spots, the little red spots. Because we have 1 million copy of this ALU element. In mouse, it's the same. The same evolution appears. The sequence are a little bit different in terms of nucleotide sequence, but nevertheless, we have something like 400,000 copy nowadays of this element. And this element was single copy 80 million years ago. So you see the increase of the number of copies that is responsible for the 10% increase of the genome size only due to these ALU sequences. Okay, this is just an example to show you that. All right. No, it's not this one. Okay, so I'm going to show you through two examples another source of genome size increase. But for now, what we saw is the increase, I mean, the origin of the increase in the size that is due to the multiplication of DNA repetition. So we saw that the human genome is 50% constituted by repeated sequence. So we have the transposon, the retrotransposon, the sign, the line, and so on, that can multiplicate, but they don't confer evolutionary advantages to the species, okay? In some cases, yes, but most of the time, no. So this is one explanation of the increase of the genomes that we see along the evolution. But there is also another source of genome size increase that have a very, very strong impact on the function. It is all that belongs to the gene family and gene duplication. So now we're going to talk about part of the sequence that can be duplicated in the genome, but those sequences, they code for proteins. And because they code for protein, it means if you have one gene that is coding for one protein and you duplicate it, it will code the same protein. So there is no obvious advantage in having two separate genes that are coding for the same protein. But nevertheless, what we can find is that with the evolution, the duplication of genes that encode for a protein can have small mutation or different pattern of expression. And at the end, it turns that you have two copies of the same gene, but which are not exactly the same. Either they can confer different function or they can be expressed in different places. And it's a very, very strong selection process for evolution. So I will show you two examples, one in Homo sapiens, and we're going to talk about the gene family of the globin protein, which is very important in mammals. And I will give you another example on plant, which is related to adaptation to heavy metal contamination. I will explain you that just after that. So multi-gene family and gene duplication. It is, as I said, a source of genome size variation, but a source of plasticity in gene expression profile, time, space. Alors, for example, in Homo sapiens, nowadays, donc Homo sapiens as we are now, okay? We've got, sorry, I didn't count them. So they are, oh, it's not written here, but next slide we see. So the two lines represent two parts of two different chromosomes. I think it's chromosome 16 and, no, this one is 16, I don't remember, I will show you. The code, all these little boxes represent globin gene. So we have a total in human of 3, 6, 7, 8, 9, 10, 11, 12, 13 different globin genes in human. There is a color code in the little box. According to the color, they explain you this copy, for example, sigma 2 and this one, eta 2 and this one, they are expressed only at the embryo stage. The blue one is during the fetus stage of the development. Red one, so alpha 1, alpha 2, delta and beta, they are expressed only when the individual is adult. And what they call phi 1, 2, and so on, they are pseudogenes. So it means we have 1, 2, 3, 4, 5, 6, 7, 8 functional genes. These 8 functional genes, 2 of those are expressed during the embryo, 2 others during the fetus stage, and 4 of them during the adult stage. The orange one are pseudogenes that are coming from duplication of the functional one, but with mutation, so they are not functional. So do you see the advantage of having a multi-gene family? So we have different copies. Those copies, they are expressed at different moments into the development. So it means that they can be transcriptionally finely tuned. And moreover, there are some genetic nucleotide sequence differences inside the coding sequence, so the function is a little bit different. So this is another way to see the same duplication of the gene. So we've got our, okay, this was chromosome 11. So chromosome 11, epsilon, gamma, gamma, delta, beta, chromosome 16, the 2 copy of alpha and this one. And what we have, the globin protein, they work as a tetramer. And so you have here different possibilities of the formation of the tetramer complex according to the stage of development. So, for example, when the organism is in the embryonic stage, the globin, the tetrameric globin complex of protein is only composed by 2 subunits of alpha and 2 subunits of epsilon, because those genes, they are the epsilon 1 and those 2 alpha are the only ones that are expressed in the embryonic stage. Fetal stage, epsilon is changed to gamma. Gamma is expressed only in the fetal stage. Adult, we've got gamma that is changed to delta, and adult, we've got minor adult, and major adult, we've got beta that is changed instead of delta. So having different copies of different genes, what does it mean? It means that, for example, the epsilon copy, the green one, it is a gene that encodes a subunit of globin, but the promoter of this gene is only active during embryonic development. Once the individual is out of the embryonic development and goes to fetal development, the gene epsilon is turned off, but the 2 genes gamma are turned on and take the place of the epsilon one. So it's really a force in terms of evolution, because what can happen there? You see that, okay, we don't look at the first one. In the 4 stage, embryonic, fetal, minor adult, and major adult, we still have all the time 2 copies of alpha subunit. But we have 2 other subunits that change. Those copies of the gene, the epsilon, the gamma, the delta, and the beta, they are different genes on the same chromosome, but they contain different punctual mutation. So the protein coming from the translation of delta can be a little bit different than the one coming from the transcription of beta. So at the end, what do we have? We have functional globins, functional tetramers of globin in all the stages during the life of the individual, but the activity can be a little bit modulated by swapping different subunits one by another one. You see what I mean? Do you understand the process? You can tell me if you don't have. It's okay. Okay, so this is another way to see the. Okay, I put this this drawing here. It's just to show you that they work in tetramer. But so here we have the time. This part on the right is more interesting. The time in million years. So 700 million years, and the phylogenetic history of the globin protein. At the beginning, there was a single protein, then an event of duplication 500 million years ago that creates the alpha subfamily and the beta subfamily. The alpha subfamily, okay, is now linked to the older version that we have in chromosome 60, and there are different alpha genes. I show you back again. Chromosome 16 is here, and we've got different copy of alpha gene. Here, the orange one, it's another one. Then after the first duplication event, alpha family chromosome 60 and the beta family on the chromosome 11. And then subsequently, there are different duplication events that give rise to all the different subunits with specificity in terms profile of expression. Okay, so adult for delta and beta, fetal for gamma and epsilon, and so on. So we can, when we analyze the sequence of these beta globin genes, we can rewrite the history of gene duplication of this gene family. Okay. So just to tell you, and after we will finish with another example, how we can have one gene or one piece of DNA that is going to be duplicated. So are you familiar with recombination or not?
Kind of. I think we have learned this in some courses before.
Recombination. You already, if we talk of genetics, the crossing overs, you know what it is?
Yes, of the clones.
Okay, so I will stop to share. I will just try to make you. I try to make you try to share my. Okay, so you see okay. 1 second. So what we have in genetics. So here I write one chromosome that is coming, for example, from the male, and this is the same chromosome that is coming from the female. Okay. Actually, I should have put it in another color. Okay. Okay. So this is for the female. So during meiosis, so I write only one. I don't write the two chromatids. During meiosis, you have exchanges of sequences between the two chromosomes from the father to the mother, and we write it like that. Okay, for example, it means that this sequence here, because it is extremely homologous to this sequence here, and this sequence here extremely homologous to this sequence, we can have this DNA during meiosis that is exchanged with this part from the mother, and a reverse way is this one that is going to be exchanged with the other one. Okay, you know that in genetics. Yes or no? Tell me if you don't know. Okay,
Yes.
So basically what we can say in genetics, it is if you have one chromosome that is coming, let's say, from the female, and another chromosome that is coming from male, if you cross them, you can have in the progeny, you can have patchwork of I show you and explain you after. You can have patchwork of genomic material coming from both of the parents. So this will be, for example, the F2. So what does it mean? It means that this sequence has been swapped with this one. So you have, so this sequence has been swapped with this one. So this means at the end, you have this that has been exchanged with this. Okay. And this is possible because the sequence that is present here is exactly the same, extremely homologous to this sequence here and here. And so we can have exchanges during meiosis. Okay. So if we look at this classical way of exchanging material in two chromosomes, you don't have gene duplication. Okay. But what can happen is this. When we have unequal gene duplication. I explain you how it is. So we have, no, sorry, I will show you because maybe it's better in the slide. You will see. Okay. So here, so do you see the pointer? Okay, you see? Okay, so we have, from what I just drew before, it's exactly the same situation, but we are not aligned vertically. So this bit of DNA is the bit of DNA of one of the parent. What happened? In this parent, it's a completely fictive situation. We've got one gene of globin, okay? But this gene of globin, on the right and on the left, you've got two sequences. Strangely, this is ALU sequence. We've got two sequences that are completely identical one to the other. Okay, so the red box is the same sequence as this red box on the left. Okay, so let's say that this line here is the father. The line underneath is the genomic sequence of the mother. The genomic sequence of the mother is exactly the same. It contains a globin gene, and on the left, you've got this sequence that is repeated on the right. Okay, same structure between male and female. But recombination happens when two sequences are identical and should be present at the same place. So, for example, we could recombinate inside the beta gene from the father and inside the beta gene from the mother. At the end, you don't have duplication because you just have exchange the right and the left. But because of the presence of these sequences that are identical and that are on the left and on the right of the gene, we can have what we call unequal crossing over. You have recombination between homologous sequences, but not at the same position. So the result of this recombination here will be, this is what is written at the bottom. One strand starts from the father DNA. We have this sequence. The father globin gene, we've got this sequence. The repetitive sequence. Then there was recombination, same sequence. Then we go for the mother globin gene, and we go back to the sequence repeated. So after recombination, you've got one piece of DNA that contains two copies instead of one and one that contains no copies. Okay, this is lethal. Okay, so you won't have any fecundation after, but at the end, from two parents that has only one copy of one gene, the progeny can have two copies because the recombination didn't have the right place. And this is a way to duplicate genes. What is very interesting in this example, but I don't want to talk to you too long about that, the red sequence is here, the sequence that are flanking the globin gene. Actually, there are sign sequence. This is L1. And sign sequence, there are repeated sequence. You see that this one is repeated here, and so on and so on. So this repeated sequence that correspond to almost 50% of the genome, one of the conclusions is that they participate to the mechanism of unequal crossing over that give rise to the duplication of genes. You see, do you understand this, this schematic? It's okay? So it's a way that the gene can be duplicated. After, what's happening here? After the event of duplication. The blue one on the left is identical copy of the blue one on the right. So no functional addition on that. But what happened is after, during evolution, you can imagine that the copy on the left is going to follow a different evolutionary scenario than the copy on the right. So some mutation can happen here, other mutation different can happen here. And at the end, starting from one single gene, you can have two genes that have subtle difference in terms of coding sequence or promoter. So you start for one gene, then you go for two, and after you can imagine that you can repeat the sequences. In this drawing, what is interesting is that the reason of this unequal crossing over is coming from the presence of L1 sequence, which is a repeated sequence, which is a kind of very old retrotransposon. So retrotransposon, they are key players in terms of genome evolution because they participate to gene duplication as well. Okay. So I give you another example, and we're going to finish here because we saw a lot of things today. It's an example on plant. Well, plants, they have a problem, but it's not a problem for them. They cannot move. Okay, so they are totally dependent on the place where they grow. So it's not a real advantage because if you have a fire in the forest, the plant, they cannot go away. So that's a problem. So they burn. But they had to develop extremely sophisticated mechanism to adapt to environment. And there are more diversity in plant for adaptation to the environment than you find in animals, a lot more. So some plants, and this plant, Arabidopsis thaliana, which is the model plant, but there are plenty of different Arabidopsis. And Arabidopsis alleri is one species of the Arabidopsis family, but it has a particular behavior that this plant is able to grow on highly contaminated soil, contaminated by heavy metals. Okay, you have places on Earth where the soil is extremely, either naturally or because of human activity, contaminated with heavy metals like zinc, lead, plenty of nickel and things like that. So if you want to grow plants in this contaminated soil, they cannot grow. But some of them manage to develop adaptation in order to be able to grow in this kind of environment. So here you have a schematic representation of a cell of three plants. So the model plant Thaliana and the model plant rice or Rhiza sativa. So model dicot, model monocot. The plant cell, they are characterized by one organite that is not present in the animal cell, which is called the vacuole, okay, which is a big organite inside the cell that is here to accumulate everything that is toxic, mainly, that the plant can absorb. So in normal plants, I mean non-tolerant or non-accumulator of heavy metal, the plant contains some transporters, and I will talk to you about this transporter. This is the blue things here. Are called MTPs. MTP means metal transporter protein. They are protein transporter that are inside the tonoplast, so the vacuole membrane. In non-resistant plant, Thaliana and rice, we have few amount of this transporter, which means that if those two plants are exposed to very highly concentrated zinc concentration in the soil, the zinc will enter the cell, but not all the zinc can be stored in the vacuole. So it means that the concentration of the zinc will increase inside the cytosol, but zinc is toxic, so the plant will die. Okay. On the other hand, you have some plants like Arabidopsis thalieri that have, because of gene duplication, they have multiplied the quantity, the number of genes, and therefore the quantity of metal transporter in the vacuole. So you see here it's represented only 2, and here we've got plenty. So what's happening is now this kind of plant, if you grow it on contaminated soil, it will, exactly as the same other, absorb the zinc, but the zinc inside the cytosol is going directly to be uploaded into the vacuole. So you see in this situation, you almost have no zinc free inside the cytosol. So it's non-toxic. Rice, so I'm not going to talk about that. Rice has another strategy, is it absorbs the zinc because it cannot do anything else. Part of it is in the vacuole, so non-toxic, and part of it is put outside the cell, rejected. We're not talking about rice. So there was a very interesting study in the comparison of Arabidopsis thaliana and Arabidopsis thalieri. So same family of plant, one with only few genes of MTP and the other one gene duplication. Thalieri is resistant to heavy metal, thaliana is sensitive. So what has been done? Zairam Shahzad in 2010 undertook the characterization of all the MTP gene family in thaliana and thalieri. So he took the two species, Arabidopsis thaliana, thaliana sensitive, thalieri resistant. The genomic data at this time, in 2010, we had the full genome sequence of Arabidopsis thaliana, obviously, but no genome sequence of Lirata, of thalieri, sorry. So here on the top, he managed to isolate. So in Arabidopsis thaliana, you see here an 80. Arabidopsis thaliana, there is a single copy of MTP, only one gene in the genome. So this is the drawing of the gene that is present in thaliana. So the drawing is like that. This is the site of transcription initiation. This is the, okay, there is an exon here, but the ATG is after. So the coding region is there on the right. So summary, thaliana, one copy. This one, you forget about it. It's, I don't know if you can read, it's called AL, MTP. This is another Arabidopsis, which is called Arabidopsis lirata, that has only one copy and the same as thaliana. Okay. These two plants, thaliana, lirata, sensitive, thalieri resistant. The two sensitive plants contain only one copy of this gene, but thalieri, which is resistant, contain now, because of evolution, 4 copies of the same gene. And this copy, they've been characterized. So first thing is they are a little bit different from the copy that we found in non-resistant plant. The promoter is different. Promoter is defined by the red part here and the boxes in blue. The promoter are different for 3 of them. One of them is almost the same. And what was found? So what is the conclusion? A non-resistant plant contains 1 copy of this metal transporter, but a resistant plant contains more than 1 copy, in that case 4 copies. And the fact of having 4 copies will increase the level of transcription and therefore the level of translation and the amount of protein at the end. So you will have much more MTP product on the membrane of the vacuole in the cell. So you will have much more uptake of zinc ion inside the vacuole. But what was found, and that was very interesting, is that so there was gene duplication. We start from 1 copy and we go for 4 copies, okay? But this gene duplication, when all the sequence were analyzed, you see those, you have the legend here. You see look at the last sentence, the retrovirus related pol protein from transposon TNT1 is located putative in the promoter of NTPA1. Here we've got traces of transposon, which is TNT, that's the name of this transposon. And actually, it's because of this transposon that from one copy in the evolution that there was unequal recombination, and at the end there is duplication, exactly the way we saw here. So imagine that this was one NTP copy, another NTP copy. Here, instead of being L1 transposon, it's TNT. And because of the presence of this retrotransposon, there was duplication. And at the end, the species Arabidopsis alleri contain now 4 copies of the same gene. And the function of this 4 copy is to be able to grow in very highly contaminated soil by heavy metal. Okay? So, okay, now we're going to finish now. Okay, so do you understand this aspect on, we take just 2 minutes and then we finish. This aspect, the relation that there is between. So I hope that you remember that in the genome, if we take the case of human, in the genome, we have something like 50% of repeated sequence. Among them, there are plenty of these repeated sequence that come from very old retrotransposon that have completely degenerated with the evolution, but this is half of the genome. And this is one explanation of the genome size increase. And then the second effect of the presence of all this retrotransposon inside the genome of this repeated sequence is that because they are repeated, you can have more and more unequal recombination, and this unequal recombination can give rise to gene duplication. And gene duplication can be very, very powerful in terms of evolution, because once you have a second copy of a gene, this second copy can have a little bit different function, like we saw for the globin. For example, the globins, there were some of them during embryo, other one for adult. So it's a paradox a little bit, which is, and I will finish with that, you increase the genome because of the multiplication of repeated sequence. But this multiplication of repeated sequence can have a positive effect in gene duplication, and the gene duplication can give to the organism new function. So it means at one moment that if you want, when you see new function that are appearing little by little, it's almost all the time accompanied by the increase of the genome, because repeated sequence are a kind of motor for gene duplication. And gene duplication is a very strong adaptative process for adaptation to the environment of new things. Do you see what I mean? Do you understand what? So at the end, at the very beginning, when the researcher, they found that there was 90% of the genome that didn't code any protein. They called that junk DNA. Junk means it's not useful at all. But actually, with what we know now, this is not junk DNA. This is DNA that comes from an evolutionary process. And this evolutionary process gives rise to new genes, new functions, so it participates positively to evolution. Okay, it's not junk DNA. It's not useless, because it's a stimulation of genome evolution. Okay, I hope it was Okay, so we finish now. I don't know if you have any question. I am
Can I
here to discuss with you. Tell me.
Yes. I want to ask because the transposon you said is the origin of the mutation. So in normal situation, this is activate, or it will be kept in silence and will be activated in some specific condition, like it can jump randomly, or is
You
yeah,
mean the activation of the transposon?
yeah.
Alors, yes. No, you're right. So it's.
I mean, I want to ask in normally it will be keep in silence, or
Alors,
it will be activated randomly.
yes, we're going to see that tomorrow and the day after. So you understand, but your question is very good because it means also, so you see that the transposon, they can jump. When they jump, they can knock out a gene. So it's very dangerous to have transposons that are active in the genome. So in our bodies, in all the living cells, we're going to talk about that tomorrow. There are epigenetic phenomenon that blocks the transposition of transposon when you are for an adult, because you don't want the transposon to jump from one place. It will be very, very dangerous. The transposition, it's a very rare event, and it happens mainly only during meiosis. Okay, this is the only moments where the transposon sometimes they jump. After, there is another situation where researchers they found that some transposons were jumping, is when and it has been especially studied in plants. For example, you take in rice, there are one major transposon is called TOS17, TOS17, the number. There are 4 copies of this one, and one of these copies jumps start to be active every time you try to make in vitro culture. Okay, so because to do in vitro culture, you put different hormones in order to start cell division and things like that. When the cells perceive this hormone, okay, there is something strange, it's the stress, and the transposon jumps. So it's a way also for the researcher to generate mutants. There is also another transposon which is extremely well studied in plants. It's called TAM, T-A-M, TAM3. And this transposon jumps at a particular temperature. If the plant grows at 25 degrees, nothing jumps. If you, for 10 minutes, you put the plant at 42 degrees, the transposon jumps. So basically, to answer yourself, most, if not all, the transposon does not move at all. Some of them move during specific stresses or at particular stage during development, essentially meiosis. And after, they are stable. It would be too dangerous. Imagine that you, in yourself, you have transposon jumping everywhere. Imagine the number of mutants you can have. So it can be very dangerous. It can lead to cancer. It can lead to plenty of things. But what we saw today, it's not in one generation. I was talking about hundreds of million years. So it's very, very, very slow. Huh? It's not very fast, this.
Okay, thank you.
Is it okay?
Yes, okay.
Okay. Anyway, what we can do, if you want, if you have things that you want me to re-explain and so on, we can do that on the next lecture. So if you have question, you can keep your question, and then I can take half an hour to answer to your question, specific question according to what you want to understand. After, I've got another question to ask you. not for tomorrow, but starting from Wednesday, do you prefer to start 1 hour before?
I'm fine with that.
Okay.
Fine.
No, it's for you because for you in Hanoi, it's from 3 p.m. to 6 p.m. Do you prefer to start at 2 and finish at 5?
It's okay.
It's up to you. For me, it's the same. I mean, I wake up very early, so no problem. But anyway, what I propose you tomorrow we do. So you have the link for the Zoom. We start at 9 tomorrow, and if you want, starting from Wednesday, that we start the lecture at 8. Tell me tomorrow, okay? And
Okay.
it's not a problem for me, okay? It's up to you. So, okay, so thank you for listening. And so if you have any question, try to write down and we can discuss tomorrow because we will go on. So tomorrow, we go on a little bit on the genome structure, but we'll talk a lot about the chromatin structure. So from that, we're going to see from the heterochromatin, euchromatin, histones. And then we're going to see the transcriptional gene silencing and so on for the expression. Okay? So if you have any question, write them down and we can discuss tomorrow. Okay?